Okay, so I want to first, I'm recording now, and I want to go ahead and look at the syllabus for things in the future here. So let's see, today is February 15th. You just had this assignment. So the first, that was only an 18-page reading of the Mastered as Emissary. The quiz for that was due at 11 a.m. just now. And I'll remind you, if you are late on any of these, I will accept them all late with really not too much points taken off at all. So definitely feel free to turn any of these assignments in late. And then we have the textbook chapter 3 quiz on Blackboard, which is due Wednesday. And someone asked at the end of last class for tips on doing these quizzes. And this is something I've heard about in this class, so I'm going to say something about it now, which is the textbook is pretty hard. There's tons of vocabulary, lots of concepts that you've probably not heard before in the readings. And so students sometimes become overwhelmed with the readings. And so a student asked last time how to approach the readings and the quizzes. And so here is my suggestion. I thought I mentioned this in the first week, but if not, I'll mention it again. I don't actually suggest you read the entire chapter for most of these chapters that you have to read. There's usually 6 or 7 like sections, and I don't necessarily suggest you read all of the sections. The way I suggest you approach these quizzes is you look at all the questions and you try and answer each question. Only read those parts of the book that are necessary, one, for you to make sure that you get the questions correct on the quiz. And then two is, in addition to just getting the answer correct on a quiz, you might want to read some of the background given that vocabulary term or the concept that a question is about to make sure that you kind of understand why the answer that is correct, why that is the correct answer. And so really, I only suggest you reading those parts of the book that pertain to the questions on the quiz. If there's a section in the chapter where I ask no questions, then you don't need to spend time reading that section. And this will become more important in future chapters when the first few chapters aren't particularly long. Chapters 1, 2, 3, and even 4 are not particularly long, but they definitely get longer starting in chapter 5. And I just want to urge you to remember, only read those sections of the book which pertain to various questions that I ask. If there's a section where I don't ask a question from that section at all, then you do not need to read that. Let's take a look at this chat thing here. Are we able to look back at the quizzes after we take them? Yes, you are. I have to make that setting in Blackboard so that you can, and I will always do that at least 1 week in advance before the test. So basically, you can retake the quizzes after. Usually, you take them, and then there's a period where you can't look at them unless you have a meeting with me. If you have a meeting with me, I'll show you your quiz whenever you want. But if you want to look at them on your own, basically, I have to click a switch on Blackboard, which will allow you to do so. And usually I do give that about a week before the first test so you can use the quizzes to study for. If you want to do that before that week before the test, simply send me an email. And I can make an exception. I can maybe put them out there like 2 weeks, or I can't do it for all of the quizzes in like that much in advance. But the quizzes like chapter 1, chapter 2, I could do those 2 or 3 weeks before the test. The thing I'm trying to prevent is students who haven't taken the quiz yet seeing the correct answer somehow and then not doing the quiz correctly because they somehow see the correct answers on someone else's. That's what I'm trying to prevent. So if you want to see the quizzes after you've taken them, simply send me an email and I can make that happen. Otherwise, I just do it on my own about a week before the test, maybe a little bit more than a week before the test. Okay, so I think I've said what I need to say about these quizzes. And I will remind you, textbook chapter 3 is due Wednesday. Today, this afternoon, I will be posting this quiz. So the next chapter, I believe this would be chapter 1. So this is the introduction for today of Master and Emissary. Chapter 1, which is called Asymmetry in the Brain, will be due a week from today. Okay, so now moving on to chapter 1 stuff. We left off here. Oops, that's a little premature. We do have an in-class questions today. Let's see. So we left off here. I'll briefly review some of this stuff. This is kind of what we were talking about mainly last time from this chapter. And these are important big picture things we'll be talking about, especially these functional methods. We'll be talking about them a lot in this class. Most chapters will include at least EEG and fMRI findings. And I'll remind you, the functional method, all of these right here, they measure brain activity, either electrical activity, magnetic activity, and both of these down here could be considered like measures of blood flow, um, metabolic activity, these two down here, metabolic activity, and they're all measures of brain activity. And these functional methods of neuroscience, of cognitive neuroscience, people are performing some type of a mental task, and we're comparing brain activity at least between two different conditions. And those two different conditions represent two different mental states or two different behavioral states. But in neuroscience, usually think of it in terms of two different, like mental cognitive neuroscience class would be like two different mental states. We're looking, how is the brain different between, how is brain activity different between these two different mental states, these at least two different conditions. You can't have, of course, more than two different conditions, but at a minimum, you have two different conditions, and the brain activity is compared between them for all of the functional methods. But we're using, once again, electricity, magnetism, and then metabolic activity or blood flow for these last two. Different ways of measuring activity depend on the functional method. These structural methods, on the other hand, these are anatomical. These methods, people aren't performing any type of task. They're basically just laying down, and pictures of the brain are being taken, and you get like a nice picture, a nice anatomical drawing of the brain. And someone asked last time, and I would agree, that these are the methods which are most useful for like seeing things like if a person has a tumor in their brain, or if a person has had some type of brain damage, or if a person has had a stroke. You can usually see evidence of that by looking at the anatomy of the brain and how it's different. You can, of course, see some of the reproductions of the repercussions of brain damage in the functional method. For instance, if a person has a lesion to the left hemisphere of their brain, or they have a tumor in the left hemisphere of the brain, and you have them perform some type of a speech task, you might see abnormal activity in the left hemisphere of the brain, the part of the brain which is often involved in speech tasks, speech perception, and speech production. So once again, structural, anatomical, and then functional is measuring brain activity between at least two different mental states, between at least two different conditions. So we talked about that last time, so I'm not going to spend too much time here. Um, yeah, I also talked about this last time. I do want to draw your attention down at this last part. Here's just some examples of some of the different mental states or some of the different tasks, the different conditions that occur in the functional method, measuring brain activity when you're looking at an upright face compared to an upside down face. Those would be the two, the angle of the face, the orientation of the face would be the two different conditions. Another example would be the type of word that you hear. You might hear a word in your native language versus a word in your foreign language. Compare the brain activity between those two. And then an example based on the shepherd rotation task, which we talked about previously in this course. There's all those different angles of rotation, the difference in angles of rotation between those two shapes. And you could compare brain activity when the angle of rotation is small compared to when the angle of rotation is big, measuring the brain activity between these are the different conditions. These are the different mental states. Brain activities compared between at least two different mental states, between two different conditions in the functional method. So now moving on to some stuff which is talked about in the textbook. And so I guess the remaining parts of this chapter are kind of like a history of the ideas. Cognitive neuroscience is relatively new compared to like psychology. Psychology itself is definitely like 100 years old, 120 years old or so. Cognitive neuroscience is probably more like 40 or 50 years old. So it's much younger compared to psychology. And a lot of that is because there's been changes in technology and the methods which have become available, which has made studying the brain more easy to do, more plausible. So part of assumption of studying the brain in a cognitive neuroscience course is this assumption of localization of cognitive function. This is just the idea that, we have this brain, which is pretty big. There's a lot going on up in the head. And the idea is that the different parts of the brain correspond to some specific cognitive function. And I've already talked about some examples of this. For instance, the left side of the brain, although it's a big, it's a whole half of the brain almost, the left side of the brain is a big region of the brain, which is strongly tied to the cognitive functions related to speech. Um, uh, the frontal lobe, which is this part in the front top part of the brain, is a part of the brain which is known to be involved with planning and thinking about the future, in addition to inhibiting impulses. And so what I'm doing, what I'm talking about here, localization of cognitive functions, the idea we have all these different regions of the brain, and we can tie specific mental abilities or cognitive functions to these different parts of the brain. And that's really what we're going to be doing this whole class. We're going to be talking about different regions of the brain. Sometimes we'll be talking about like big regions of the brain, like entire hemispheres. Sometimes we'll be talking about like small pieces of the brain, like the hippocampus, which is this region of the brain. It's kind of in the middle of the temporal lobe, or the amygdala. There's different examples of brain regions which are small that we'll be talking about, but we'll also be talking about bigger brain regions. But throughout this class, this idea of localization of cognitive functions, this idea that we can, there's some specific cognitive function which is tied to each of these brain regions that we are studying. That's the idea of localization of cognitive function. I want to go through this stuff right here quick because I don't usually ask too many questions about it on test one, but I don't want to completely skip over it. It's kind of just a history of this idea of localization of function. The idea is that people have been kind of assuming that the brain and the head are important for mental states for a long time. And it's actually started out with as old as ancient Stone Age people, so, thousands of years ago. Some of the evidence that they knew that the head was important for thinking. So that's kind of a form of localization. And they're saying they weren't pinpointing specific regions of the brain, but they knew the stuff up here in the head was somehow important for thinking. Because this idea of trephination, people that had abnormal thoughts, or maybe who are having lots of headaches, but abnormal thoughts is the one which is most relevant to the idea of thinking or mental states. They would use trephination, drilling of the holes in the skull, and was supposed to be some type of therapy. So there's presumably archaeological evidence in support of this. So, it's the localization because they think abnormal thoughts were coming from the head. Their therapy didn't really make sense, drilling holes in the head, but they were at least in the kind of the right general vicinity of, they're starting to localize mental activity to this region up here. Moving on. Ancient Egyptians, I'll move that picture of Jim Carrey. They continued on with this idea of localization. They kind of knew from watching people who are injured in war, that if you have an injury to the head, if your head was injured, that it could cause things like motor impairments, and it could also cause problems of the ability to like speak. And it could also cause obviously problems in consciousness. So these individuals were connecting different mental traits. Once again, they were still kind of close to the head, even though they weren't able to localize it to specific parts of the brain yet. They knew that the head was important for these different behavioral and mental states. An interesting thing about the Egyptians is that they also were interested in the role that the heart might play in different behavioral and mental states. And so this is kind of an interesting twist. We usually think of the brain as being the part which is important for these different mental and behavioral states. But some people, and it's not only the Egyptians, also localized some of these functions to the heart. They said the heart might actually be important. And that's not, it sounds kind of crazy, but there's clearly some type of link between the brain and the heart. And just to illustrate it very simply, obviously different emotional states have some influence on your heart. For instance, when you're like angry, for instance, that can cause your heart rate to increase. There's definitely clearly some link between emotional states and physiological states, including the rate at which your heart beats. So the link between them is not still clear even to this day. They don't really know what is the relationship of the heart to these different mental and behavioral states, but there clearly is something going on, and ongoing research is trying to understand what is the heart doing for these different mental and behavioral states. We know that the head is important, but the heart obviously the heart appears to be doing something as well. And, ongoing research is trying to figure that out. Still in this idea of localization, the idea that there's these different parts of the brain, and we're localizing cognitive functions to these different parts of the brain. And this idea still, now we're just moving through history, and the Greeks, that's still, a couple thousand years ago, a little more than a couple thousand years ago. But they were kind of doing similar, ideas as, the Egyptians before. They just moving along in the future, getting a little bit more complex. Um, Hippocrates, oftentimes thought to be like the first medical physician, suggested the brain, the organ inside the head, is important for perception and emotion and different aspects of conscious experience. And so this is a kind of a new little twist on things. He talked about body fluids or humors can disrupt normal behavior. So this is not the term we use or that neuroscientists use, but this idea of fluids within the body or within the brain that are important for behavior and mental states, that's relevant. Now it's these types of things would probably be known as neurotransmitters or hormones. But that is some that low cognitive functions can to some extent be localized to the activity of different chemicals or neurotransmitters in the brain and in the body. So they're making progress here by this idea, even though they called it something different than what we call it today. I'm actually going to skip over this stuff right here. And I know I don't happen to ask any questions about that on test one, so I'll skip over these last two bullet points. And all I really want to say about this Descartes stuff, um, I mean, he was doing localization of function, I guess I should say. He talked about this one region of the brain called the pineal gland, which is kind of in the middle area of the brain. And he thought that this was a region of the brain where like the mind and the body, these two separate things, were integrated. That was like he localized the integration of mind and body to this one specific part of the brain called the pineal gland, which today there's really not that much evidence that this is true. From my understanding, the pineal gland, this brain region that Descartes, another philosopher who's around the 1700s, he was really interested in the pineal gland. It seems to be most important these days for helping to regulate like sleep and wakefulness. And it releases melatonin, which is a hormone which is most famously known for making people tired. So this visual topography stuff, I'm going to skip that for the sake of time because I happen to know I'm not going to ask questions about that on test one. I do want to talk a little bit about this idea of rationalism versus empiricism. Descartes would be considered a rationalist. And this idea, I think I have some stuff here. Yeah, I have the terms down here in the notes. Rationalism is the idea that you can learn about the world from just like thinking about it. You can just use your mind to understand the world by kind of philosophizing or using your intuition. Compared to empiricism, empiricism is like what the current scientific method uses, which is the idea that we use observation, specifically like, seeing something occur, using your body, like your eyes, usually is the way this empiricism comes from, using observation. You can think of it as seeing something to learn about the world. And so the key difference between rationalism and empiricism is rationalism is completely mental. It's all in your brain, supposedly, all in the mind part. Where empiricism, using observation to learn about the world, you're somehow incorporating your body. That is, observation requires your eyes, which are part of your body. So the distinction between these two terms I could ask about on test one. Okay, so we've been talking about localization of function. I wanted to talk about kind of the opposite idea, aggregate field theory, which is, there's a different term for this now, but this is a term this concept is still relevant in modern cognitive neuroscience. So aggregate field theory is in some ways the opposite of localization. That's why I say early opposition to localization. Localization is the idea that you can pinpoint different anatomical regions, usually of the brain, but sometimes of the heart, as some people did, different cognitive functions to these different parts of the body, different parts of the brain. Aggregate field theory is kind of the idea that we actually can't, we might not be able to do that. There are some cognitive functions that are inherently distributed among multiple regions of the brain or multiple parts of the body. And this is based on this researcher, Florenz, who suggested that different things like sensation and perception, I haven't really defined these two terms. Sensation and perception basically have to do with using your senses, your eyes, your ears, your nose, your mouth, and your sense of touch. And perception is kind of a related term. He suggested that these were all kind of, and all of these different things, including memory, sensation, perception, and memory, were all kind of part of the same entity, or he called it, or at least the term from the book is faculty. And he could not localize these different things, these different cognitive functions, sensation, perception, memory. He could not localize them to one specific brain region. And so Florenz hypothesized that these different cognitive functions are kind of distributed among multiple brain regions simultaneously. And if you lesion one brain region, if you destroy one region of the brain, there are still other brain regions which are kind of involved with these different cognitive functions. And so everything kind of stays okay, even if one brain region is kind of damaged, because there's multiple brain regions which are subserving different cognitive functions. At least these specific cognitive functions right there involve multiple brain regions. So hopefully you can understand the difference between localization and aggregate field theory. Localization says you can kind of pinpoint a single area of the brain for a cognitive function, whereas aggregate field theory says that it's multiple regions of the brain are supporting a single cognitive function or these different cognitive functions. And as I said, some of the early research from Florence is based on ablation studies or lesion studies, which is basically where you destroy part of the brain and then you see how the animal's behavior changes. And he came up with this idea of aggregate field theory because he could destroy a single area of the brain and it would appear that the animal's memory, sensation, and perception were still intact. And so it was very difficult to destroy a single area of the brain that would impair all of these different cognitive functions. Moving on. So I'm still going kind of in time. And so we're going back and forth between ideas that support localization, then aggregate field theory. And then here's another idea which supports localization. These different competing schools, localization and aggregate field theory. This individual, Jackson, provided some pretty good evidence once again for localization. And I will mention localization is kind of the underlying assumption of this class. It's cognitive neuroscience class. We're trying to localize cognitive functions for the most part, specific parts of the brain. And Jackson definitely is one of the early people to do that. He noticed right hemisphere lesions, that is damage to the right side of the cortex, were more likely to disrupt spatial processing compared to the left. So spatial processing is like if I put stuff out there on space and it appears and then it disappears, it's like being able to track where those different things in space occur. That's like an example of spatial processing. A related spatial task would be like the Shepherd rotation task, where you actually have to represent shapes visually, and then you might have to turn one of the shapes in space, in a three-dimensional space. That would also be considered a spatial task. And there's some evidence from Jackson that when individuals, their right hemisphere was lesioned, the right hemisphere was damaged or ablated, that was more likely to impair these types of visual spatial tasks than if the damage was in the left hemisphere. So that suggests that you can localize to some extent spatial perception, or those aspects of the mind involved with processing space to the right side of the brain more than the left. So the right side, you can localize spatial processing to the right hemisphere to some extent. He also localized this thing known as the Topographic Motor Map, which we'll talk about in this chapter. And I noticed we talk about this kind of throughout this course in different chapters. So, and I'm pretty sure that there's questions about this on at least one of the tests. Topographic Motor Map. Jackson, same researcher, noticed it's kind of an interesting finding that he would observe individuals when they're having seizures. And so seizures are basically when people, their muscles will jerk and they'll kind of have these violent shaking episodes. He noticed that when individuals had seizures, there would oftentimes be a sequential pattern or an organization to the nature of the muscle movements that would occur, where one set of muscles would start to move at the beginning of a seizure, and then the muscle groups that would jerk, that would shake, would kind of start in one muscle group and then travel to another muscle group within that individual's body and then to a muscle group. So I don't know the specific order, but you could imagine it's like a person's, head starts shaking and then that stops, and then their hands start shaking and then that stops, and then their torso starts to shake and then that stops, and then their legs start to shake and then that stops. So it's like this movement of the muscle movements throughout, from one muscle group down to another, and it occurs over time during a seizure. And so he hypothesized basically this idea that there must be a part of the brain, which he called a topographic motor map, where the different parts of the body are represented. And that when you're having a seizure, what's going on is that the messages are traveling from one part of this brain, where one set of muscle groups are represented, and it's traveling down to these other regions of the brain. And as the seizure is traveling down these parts of the brain, the different muscle groups on the body start to jerk, they start to shake. This different seizure starts one part of the body, and as it travels down the brain, it's traveling to different parts of the body as well. So there's this topographic correspondence such that, according to Jackson, that one part of the brain represents one part of the body and another part of the brain represents a different part of the body. So we're localizing still. This part of the brain represents motor movements in one part of the body. This part of the brain represents motor movements in a different part of the body. This part of the brain represents motor movements in yet a different part of the body. That is the topographic motor map. We will talk about more about that. In fact, yeah, right here, we're going to talk about it right. So this is Penfield, another researcher who came a little bit after Jackson, who was a medical doctor. And he was able to do some pretty interesting surgeries. So he would do procedures. He could do brain surgery, and the people would be awake while they're having brain surgery. And so, he would use a strong local anesthetic and then would actually open up people's skulls and could see inside their brains. And he would use something known as the Montreal Procedure, which is basically a way of electrically stimulating the brain. So the Montreal Procedure has kind of like two functions. The original function was to help localize seizures, to help figure out where seizures are coming from. The idea is that if an individual has seizures, and then you stimulate the part of the brain with electricity, which is causing the seizures, you could cause that individual to have a seizure. So that's the main original use of the Montreal Procedure. But the other thing that the Montreal Procedure can do is, assuming that if a person does not have, if there's a part of the brain where a person does not have a seizure when you stimulate it, then you can actually just turn on that part of the brain. You electrically stimulate a given part of the brain. You can actually kind of temporarily turn on that region of the brain while you're electrically stimulating it, and then it turns off, a little bit later. Penfield found evidence that supports this idea. Once again, Jackson had this idea, topographic motor map. Different regions of the brain represent different parts of the body. Penfield basically found evidence using the Montreal Procedure. He was stimulating parts of the frontal lobe right here. So the frontal lobe is all of this stuff right here. And then the tail ends, this is the posterior or back end. This is all the frontal lobe right here. This orange thing right here, this is the tail end or the back end or the posterior end of the frontal lobe. Penfield was stimulating this with the Montreal Procedure. He was stimulating different parts of the posterior parts of the frontal lobe with electricity. And what he noticed was a very similar phenomenon. He wasn't causing seizures in these individuals, but what Penfield noticed is that if he stimulated one part of the brain, like up here, he could cause the individual's hip to move, or the shoulder to move if you stimulated this part of the brain of the frontal lobe, or the elbow. And there's this, like, sometimes known as a homunculus. You might have learned about this in intro psych. I know I teach about this a little bit in intro psych. These are the different brain regions of the primary motor cortex, this orange part, the posterior part of the frontal lobe. And as you stimulate these different areas, and so you start like up here and then you travel down this direction, as you stimulate different regions of the primary motor cortex, the posterior frontal lobe, you can cause different regions of the body to shake. You can have cause little movements of these different parts of the body. So this supports the idea of localization. Motor movements of different body parts appear to be represented along a continuum, sometimes known as a homunculus, on this orange slice right here. Stimulating different parts of this slice of the primary motor cortex, the posterior frontal lobe, can cause different parts of the body to move. There is a related idea called the primary somatosensory cortex, which basically says the exact same thing, but that would be stimulating the blue part right here, which is actually technically part of a different lobe, the parietal lobe, which is all this stuff back here. So this blue stuff is the front or anterior part of the parietal lobe. The idea of the primary somatosensory cortex is that you can stimulate different parts of this blue strip, and you don't cause motor movements, but you can cause illusory feelings of those different parts of the body. So I stimulate right here, and I could cause a person to feel something occurring in their neck or their head. I stimulate right here, and I can cause the person to feel something in their arm or elbow. Down here, they could feel something in their face, down here in their teeth, so on and so forth. Whoops. So primary motor cortex, you can actually cause different motor movements by stimulating different parts of the brain. And primary somatosensory cortex, you can cause a person to feel different sensations, different feelings, depending on which part of the brain. In both cases, it supports this idea of localization of function. Different parts of the brain are representing, in this case, different motor movements or different feelings, the different regions of the body, different feelings, different motor movements of different parts of the body. Okay, so let's, how we're doing on time here. Not bad. Okay, so now we're going to move to, we've been talking about kind of some of the big picture stuff in the brain. We're talking about like the, big parts of the brain, like the left and right hemisphere, for instance. We're going to move for a while, actually, towards the end of this chapter and then into chapter 2. We're going to be talking a lot about the small parts of the brain. So the thing which is challenging about the small parts of the brain is very difficult to say, for instance, that this is a nerve cell or a neuron right here. It's very difficult to connect mental states to the very small parts of the brain. Usually mental states in cognitive neuroscience research are connecting a big region of the brain to a mental state. But for a while, the next couple chapter, or the end of this chapter and then chapter 2, we'll be talking more about the small scale of the brain, which is challenging for cognitive neuroscientists. It's difficult to connect the small scales to different mental states, but we're still going to talk about this stuff a little bit in this class. This class definitely, I'd say 90% of this class is about kind of the macro, the big parts of the brain, but we will talk about the small parts. So here's a neuron, a nerve cell. You probably saw one of these in introductory psychology. If not, we'll still kind of review the same stuff that you would have seen in intro psychology. The idea of these nerve cells is that they convey messages from one nerve cell to the other. You can kind of think of nerve cells or neurons as being connected in like chains, and that one side of the chain or one side of the nerve cell receives a message, and then it conveys that message to the other side of the neuron, and then it can pass that message on to the next neuron, the next nerve cell in the chain. And so here's a single nerve cell. It both has a receiving part of the neuron, which is known as the dendrites. I always think of dendrites. These are like little hands that are kind of being stuck out, and they're like waiting for something to be put in the hands. That the specific thing they're usually waiting for would be a neurotransmitter, a chemical. But I always think of them waiting for like a high five or waiting for something to be dropped in them, and they receive. The dendrites are the receiver. They're hands that are waiting to receive. They're receiving messages, to be specific, neurotransmitter, chemical message usually. The nucleus, the next part of the neuron. For our purposes, we'll just think of the nucleus. This is where the genetic material of the cell and organelles within the cell are located. So there are a lot of important genetic things which are occurring. We'll talk about them a little bit, but we're not going to focus heavily on the genetics in this class. But there's definitely some important things. I'd say modern neuroscience research has become more interested in genetic changes which occur during learning in particular. So the nucleus, though, genetic information and organelles of the neuron of the cell located here. The axon right here, this is an important part of that communication I was talking about. The neurons receive chemical signals in the dendrites right here. And then if enough chemical signal is received, then there can be something called an action potential, which occurs. An action potential is where electrical activity starts right here, and then it travels down the length of the axon. I kind of think of the axon as it's kind of like a neck. It's a long neck, but electrical activity would start about right here at the top of the axon and travel throughout its length until it reaches the very end, which is called the axon terminal. So axon is just this long thing. Electrical activity of an action potential travels along this. And at the very end is the axon terminal. The terminal just means end, axon end, which is where neurotransmitter could presumably be released once again, and then it could be received by dendrites on the subsequent neuron in the chain. The last thing to talk about here is this Myelin sheath, which is basically, it is composed of glial cells, a specific type of cell in the brain and in the nervous system. And the main function of the myelin sheath is to speed up electrical transmission. You have this electrical signal which is traveling down the length of the axon, and this myelin sheath allows the electrical signal to kind of make jumps. And if you have a good solid myelin sheath like this one right here, instead of the electrical activity having to travel along the length of the entire axon, the myelin sheath allows the electrical signal to make these jumps like this, like what I'm doing with my arrow on my mouse, which in effect allows the electrical signal to travel down the axon more quickly. A couple more things about neurons. And then, so these are two individuals, Brodmann and Golgi, and they studied kind of the microanatomy. And so Brodmann, you actually hear about that. If you read fMRI papers, they always talk about Brodmann area, and then there's all these different regions of the brain, Brodmann area 17, Brodmann area 18, 39, 40, 5, 7, 4, 6, all these different areas. And it's a little bit confusing. I'm not a huge fan of this, of using the Brodmann coordinates, because these different regions don't really have, for the most part, any clear cognitive functions which are tied to them. The different regions of the brain were basically somewhat randomly chosen by Brodmann. And what he did was he would just study these different regions of the brain anatomically. He would take a neuron, a specific neuron for Brodmann region 21, and then he would look at it up close under a microscope and study its anatomical structure of neurons in Brodmann area 21. And he would do that with each of these different areas. And it kind of says that here. He studied the cell structure of these different regions of the brain. So these were chosen somewhat randomly. And he was just looking at the microscopic structure of neurons in each of these different regions. But I just want to emphasize these different Brodmann areas do not necessarily correspond closely to different cognitive functions. So it's not like each of these regions represents a different function. These are the different regions that Brodmann happened to choose to study the microscopic structure of these different parts of the brain. Golgi is another individual, and these pictures are taken, I believe these are taken from your textbook. Golgi had this idea of syncytium, and you can kind of see it here. There's a bunch of neurons here, like 1, 2, 3, 4, 5, 6. Looks like 6 or 7 neurons shown here. So we're still looking, of course, at the microscopic structure of the brain, but instead of just looking at a single neuron here, Golgi developed these staining methods, which allowed researchers to basically look at the way neurons were connected to other neurons. And this idea of syncytium, I think I have that defined here. Yes, syncytium, continuous mass of tissue with shared cytoplasm. Kind of a wordy definition, but I just want to notice that this definition of syncytium, it kind of has it's kind of like aggregate field theory. I see that question. I'll get to it here in a second. Aggregate field theory is this idea that cognitive function is distributed among multiple brain regions simultaneously, that you can't, aggregate field theory says you can't localize cognitive functions or behavioral functions to a specific region of the brain. And when you think about this idea of syncytium, basically what it's saying is all of the neurons that compose the brain are always connected to other neurons in the brain. Everything in the brain is connected as though the brain is one giant entity. It's like a single entity, or it itself is a single giant cell composed of a bunch of different pieces. And if you think about it in those ways, the idea of aggregate field theory kind of makes sense because everything in the brain is connected. There really isn't separation into different little clusters of brain region. Brain region does this, brain region does this, brain region does this. Everything's connected. So according to the idea of syncytium, it's a little bit difficult to kind of categorize or put into different components of the different parts of the brain. So it kind of, syncytium kind of is consistent with the idea of aggregate field theory. Let's take a look at this question here. Yes, the question is, did Golgi also study Golgi apparatus? Yes, I believe that is the case, I did study that in biological mechanisms. That was L201 back at Indiana University, literally back in the year 2000. So please don't ask me what the Golgi apparatus does. I actually do not remember. It was 21 years ago. But yes, I did study that once. And Golgi, I'm pretty sure it's the same Golgi that studied the Golgi apparatus, also studied the microscopic structure of neurons. I'm almost done here with chapter 1. We got another question. So the question is, I think this is a question about syncytium. So would there be a large cell membrane stretching a long way throughout the brain? Literally, no. That is not literally the case. You could think of that idea. Maybe I should go back to this. the idea of sensitium is more of like a metaphor. It's not saying that the brain is literally one giant cell with the cell membrane, one single cell membrane, like encapsulating the entire brain. It's more of like a metaphor. It's saying the brain is kind of like a cell in the sense that in a single cell, it's like a single entity, and all of the different parts of the cell are working together for that one thing. And it's kind of saying like a metaphor. The brain is similar to this. The brain is like one thing and has a whole bunch of different parts, but all those different parts are always working together. They're always connected. And so we can't really partition the brain off into these different parts, partition into different parts. You can't do that. Okay, good. I'm glad that helps. That's the idea of sensitium, kind of like aggregate field theory. Just a couple more things. I wanted to get started on chapter 2. I just don't think we're going to have too much time for that. Maybe a tiny bit. Okay, so this is connecting this idea of sensitium or aggregate field theory to kind of its modern instantiations, to some modern ideas. Some of these you might have heard about, but the kind of the key concept nowadays is that I see a lot of this like a neural network. This is actually taken from a author who's kind of famous for studying the structure of neural networks. And the idea, according to this idea of neural networks, is kind of consistent with the idea of sensitium, that everything in the brain is connected, but there's some order, there's organization to the way that these connections occur in the brain. And you can kind of see, this is a very simplified, the brain has tons and tons and tons of neurons. And what we're showing, this is just simplified neural network that, it has 4, 5, 6, 7, 8, like 10 neurons. So it's just a very, very, very small sliver of the brain. And it's basically saying this is potentially what neural networks in the brain could look like, although a very simplified neural network. And the idea is that connections in the brain aren't just random. The brain, everything in the brain is connected, but not everything in the brain is equally connected to, there's certain cells, certain neurons in the brain, which are highly interconnected with groups of cells that are close to them. And then there's another region of brain where those neurons right here, they're highly interconnected with other neurons around them. In general, neurons tend to be more highly connected to other neurons, which are adjacent, that are next to them. These neurons are close to other neurons next to them. These neurons are close to other neurons, which are next to them. And that's what we're seeing here. This is like one community. This is like one part of the brain. And this, like these neurons right here, you can see there's a lot of lines between them representing information connections, the ability to transmit signals between these neurons right here. And then here's another community. Once again, you can see that these neurons over here, they're able to kind of pass messages amongst each other. However, the whole brain is a sensitium. Everything is connected. And you can see that right here. There's this like hub neuron right here, which allows the information that's represented in this community to be transmitted to information in the other community. And so if you can kind of visualize this neural network, this kind of modern idea allows for the idea of localization of function, but it also allows this idea of aggregate function at the same time. You can localize function to communities. These are neurons which are highly integrated with other neurons that are close to them. So that's the localization part. And right here it says a concept is theoretically represented among a community of neurons. So you can represent a specific mental or behavioral, some type of a thought or some type of a behavior or some type of cognitive function to the activity of one community, and a different behavior or a different cognitive function to a different community. So this is the idea of localization. Each community can do something different based on the fact that those neurons in that community can communicate with other neurons in that community. But the brain overall is aggregate. It is a sensitium in the sense that all of the information here can be transmitted to all of the information here. So if you kind of look at this, you can see this type of a model, which is kind of the way things are going in modern cognitive neuroscience. It's consistent with localization of cognitive function, but it's also consistent with this idea of aggregate field theory or sensitium, the idea that everything in the brain is connected to everything else. And this stuff, yeah, there is a question about this, I believe, usually on the test, so I don't want to skip over this. Yeah, historically it's important. We're about to move into, in the next chapter, we'll definitely, in subsequent chapters, definitely be talking more about connecting cognitive functions to different regions of the brain. That'll especially start in chapters 3 and 4. Behaviorism was this idea that kind of preceded cognitive psychology and cognitive neuroscience. You probably learned about this in intro psych. Some, let's see, classical conditioning, operant conditioning were concepts which are very relevant to behavior, were very relevant to behaviorism. Behaviorism is basically the kind of the underlying philosophy of behaviorism, which is completely the opposite of what we're doing in this class, is that we actually cannot, according to behaviorists, we cannot study the mind. We cannot even really study the workings of the brain. And the reason is because we can't see what's going on inside the mind. We can't really see what's going on inside the brain. And if we're trying to be empirical, if we're trying to observe stuff, then we can't study what's going inside the mind and the brain because we can't observe it. We can't see. We can't use our bodies, for instance, our eyes to see what's going on in the mind or brain. Therefore, it's kind of scientifically or empirically off limits. But, as we'll see, and we already really kind of saw evidence of this, I showed you the Shepherd rotation task early on. That was an early example of cognitive psychology. Cognitive psychology is this idea that, no, we can study the mind. We might not be able to see inside the mind per se, but we can make inferences about the mind from measuring people's behavior. We can look at their behavior and make inferences about what they were doing in their mind. And we saw the classic example is Shepherd rotation task, which I showed you previously. And this idea of cognitive psychology, that we actually can make inferences about the mind, although we're not looking directly at the mind, we can look at the body, make inferences about the mind, kind of started, at least people say, according to this guy, Noam Chomsky, who I actually knew this guy because a punk band I used to listen to, I still kind of do, but their recent albums are not very good. NoFX, they have a song where they talk about Noam Chomsky. The thing I first heard about him is he had kind of some political philosophy. That's how I first heard about him. But then it actually turns out that he was kind of a linguist, and he was also basically is the person who's kind of responsible or said to be responsible for the creation of cognitive psychology and therefore cognitive neuroscience. And the textbook says that he was very important for creating cognitive psychology, cognitive neuroscience. Basically, I'm not going to go into the details of this, but basically behaviorists were trying to explain how people learned language. And they were trying to do it without talking about the mind. Because behavior said we can't talk about the mind. We can't see it, so we can't talk about it. But they were trying to explain how we learned language. And basically what Skinner was saying is these people are trying to explain the acquisition of language and speech without talking about the mind. But if you really listen to what they're saying, they're talking about the mind constantly. They're pretending that they're not really talking about the mind. But if you really look at what they're saying, they're just constantly talking about the mind, but they're just using complicated and sophisticated jargon to kind of pretend that they're not talking about the mind. And that's what I'm saying here. He basically said, these behaviorist principles to trying to understanding language acquisition were kind of loose, and they were symbolic or metaphorical, and they weren't really well operationally defined. Basically, he was saying it's BS. I have read the paper where he does this, and it's a pretty famous paper where where he basically is creating a philosophical argument, kind of destroying behaviorism. And as a result, cognitive psychology was thought to be born because of this one paper that Chomsky wrote, which criticizing the way behaviorists explain language acquisition. And that's kind of important for us. This is a cognitive neuroscience class where we will be studying the mind and the brain and how they're kind of integrated. Okay, so that's all I'm going to actually get to today. I think I want to give you some time for this. We will definitely start chapter 2 next time. We're still a little behind the syllabus, but not so much that I'm overly concerned. All of the assignments as they're currently listed in the syllabus will keep on going with those due dates as they're listed, though the lecture part of the class, tiny bit behind. So I want to point out that I updated the slides for chapter 1 because I've added a question right here. These questions were already in the in-class discussion quiz for chapter 1 that was posted last week, but I re-uploaded the slides because I wanted to add a question. This question comes from the stats research method stuff that we did last week, and I don't want you to completely forget some of this stuff, so I've added a question. So I'll kind of go through these questions. Question 1 is about the Shepherd rotation study, which we've talked about today. We've talked about it previously. And then structural and functional neuroimaging, I talked about that today and last time. Localization and aggregate field theory, we definitely talked about that today. And then I also, for this last question, number 4, I want you to calculate marginal means for the main effect of diagnosis in the graph shown right here. So this is a graph that I showed you in the stats research method slides, and I just copied and pasted it right here. And so it might help if you, and it almost certainly would help if you kind of go and make sure that you remember what a marginal mean is from stats and research methods, that part we talked about earlier. And I asked you to calculate the marginal means for the main effect of diagnosis for this data right here. And when you're doing this, you can assume that the sample size for each of these 4 conditions are equal. That is, there's the same amount of people here, 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 and here. The sample sizes across the 4 conditions are equal. And I want you to calculate the marginal means for the main effect of diagnosis. That is what you'll be doing for chapter 4. And I just, to give you a hint for this one, we did a similar problem for this example where we calculated the marginal means for the main effect of diagnosis. We've actually done that. This was a 3x2 example right here that we did that for. 3 levels and 1 factor, 2 levels for the other factor. The example for today's problem is a 2x2 design, but you're still calculating the marginal means for the main effect of diagnosis. So you've done this before. I just want you to do it for the graph that I just showed you, which is a slightly simplified. It's even more simpler than this problem because it's only 2x2 instead of a 3x2 design. So that's it. That's all we're going to get to today. I will turn off the recording now, but I can definitely stick around and answer questions.